Hello and welcome to your Chapter 11 video lecture series. We are going to talk about meiosis and sexual reproduction. Starting with section 1 on the process of meiosis. So I want you to keep in mind that meiosis is only happening for us to produce gametes. If you remember in class, we talked about gametes being an egg or a sperm cell. So that means either the ovary is producing it or the testes, depending on what organ you have in your body. And it will create genetically different haploid cells. Remember, the word haploid means that we only have 23 chromosomes in each of the cells. So meiosis 1 is going to develop two haploid cells. So we'll have each one of these haploid cells containing one of those sister chromatids that we talked about in mitosis in chapter 10. Whereas meiosis 2 will separate the sister chromatids into 4 haploid gametes. So if you're still a little confused at this point, don't worry. We are going to show some visuals and talk through it. So our learning objectives for this section are to describe the behavior of chromosomes during meiosis and the differences between the first and second meiotic divisions. Then describe the cellular events that take place during meiosis. Explain the differences between meiosis and mitosis, and explain the mechanisms within the meiotic process that produce genetic variation among our haploid gametes. Again, gametes are referring to the sperm cells or the egg. So first, let's talk about sexual reproduction. As I mentioned, gametes are the sperm cell or the egg, right? Notice how both of these are haploids and only have 23 chromosomes. And they are going to come together. The sperm cell really kind of swims up toward the egg, and there is some enzymes being released. We'll get more into the details of how this happens in A&P, but essentially the sperm cell will enter the egg, and then its genetic information will come together with the egg's genetic information, and we will have a zygote, which is our fertilized egg, and then we will have our 46 chromosomes. So at this point, it is a diploid. So these are our gametes, but what we talked about in mitosis were really regarding our somatic cells. They already have diploid chromosomes and are known as homologous chromosomes. Remember, we saw that karyotype with all of the chromosomes kind of lined up, and we saw the matched pairs that had the same height. They're going to have the same genes at the same locations or loci. So let's talk about meiosis. Meiosis is when we're going to have a diploid parent cell produce haploid daughters, and we split this up into two divisions. We have meiosis 1 pictured here and meiosis 2. Remember that mitosis, not these processes, produce daughter cells whose nuclei are genetically identical to the parent nucleus. And in mitosis, both the parents and the daughter nuclei are at the same ploidy level, which is diploid. We are going to see that this is different through the full process of meiosis. When we have our first round of meiosis, we are going to take this diploid cell and end up with two haploid cells. Whereas when we go into meiosis 2, I like to think of this as being the most similar to mitosis, because we are kind of copying, or I shouldn't say copying, but dividing this parent cell. And we still end up with haploid cells down here. So let's talk through meiosis 1. This is going to be preceded by interphase, which we saw in the previous slide. If I go back we can see interphase over here, and we know that interphase consists of our G1, S, and G2 phases, where our cell is growing, we're replicating the DNA, and we are going to basically prepare our final steps for meiosis in this case. So it's pretty similar through the interface that we go through for mitosis. Now let's talk through the different phases of meiosis 1. So our first phase is prophase 1, and yes, there's a lot that happens in here. So we're going to go a little bit slow through this. During this stage, we are going to have our homologous chromosomes, meaning pairs of the same chromosome from each parent. So we can think of this as being, let's say, the mother parent and this being the father parent. And they'll first attach to the nuclear envelope at their tips with the help of some proteins. So they're attaching to that nuclear envelope. But just as we saw in mitosis, we'll start to have that nuclear envelope break down. And so additional proteins are going to be used to pull these homologous chromosomes closer together. Now, one important feature that we can kind of see pictured over here is going to be our synaptonemal complex. And this is a protein structure that's aligning these homologous chromosomes side by side at precise locations along their length. And this alignment is allowing for synapsis to take place. Okay, synapsis. And that is going to be a tight pairing of these homologous chromosomes and really ensures that genes from each chromosome is going to be lined up correctly with their counterpart, setting the stage for genetic recombination. So the key event really in prophase 1 is crossing over to take place, which is also called recombination. And this is when the non-sister chromatids, one from each homologous chromosome, is going to physically exchange segments of DNA through the process that is guided by our recombination nodules. And these are just some large protein assemblies that are going to be positioned along the synaptonemal complex that we see pictured here. I wish we had a picture that combined these, but we don't. So there are two separate things, but working together. And as we can see here, the crossing over is going to produce recombinant chromatids that we see pictured over here, meaning that these chromatids have new combinations of their alleles alongside the non-recombinant chromatids. So see how these have been kind of untouched. After this exchange, the crossover points are called chiasmata. And really the result of this crossing over gives us genetic variation. This is why we can look so different from our siblings, since each gamete will carry a slightly different combination of alleles than the parents do. So let's continue with prophase 1, and I do have some images here that are walking through some of the steps we've already talked about, but I want you to think that even though the X and Y chromosomes are not completely homologous, they can still pair up during this stage. That's important for proper segregation that we're going to talk about later on. So after the crossing over has occurred, our synaptonemal complex, remember the protein structure that's holding our homologs together, begin to break down. And at the same time, our chromosomes are going to start to condense further. That makes them more visible under a microscope. And although our synaptonemal complex is gone, the homologous chromosomes are going to stay attached through two key places. And it's kind of hard to see in this small image, but we've got our centromere and the chiasmata that we discussed earlier, which are those physical crossover points. And if you remember those cohesion proteins that were helping hold the chromatids tightly together, they are partially removed. So at this point, by the end of prophase 1, the only thing that's really keeping the homologous pairs connected are the chiasmata. And now we are going to see that each homologous pair has 4 chromatids, and we are going to call this a tetrad. Now let's move into pro-metaphase 1. And during this stage, we're going to see that the spindle fibers, which, if you remember, are made up of microtubules, are going to begin to attach to the chromosomes. And specifically, they connect to those kinetochore proteins at the centromeres. Now you could see this also in the bottom diagram over here, and this is kind of a close-up of that centromere and the kinetochore and those microtubules attaching here. And we need to understand here that the microtubules from one pole are going to attach to one chromosome in the tetrad, while microtubules from the opposite pole are going to attach to the other homolog. So we could see that they're coming in from opposite sides. And this ensures that when the separation happens later on, each homologous chromosome will move toward the different pole of the cell. Notice that in pro-metaphase 1, the sister chromatids of each chromosome are still fused at the centromere, and they will not separate just yet. So I think that's better depicted over here, though. And then let's move into metaphase 1. Here we start to see that the homologous chromosomes are going to line up along the metaphase plate in the center of the cell. But what's really important is that this alignment is completely random. So each pair of the homologous chromosomes can orientate with either the maternal or the paternal homolog facing each given pole. And this randomness introduces a second major source of genetic variation in addition to the crossing over that we had already discussed. So remember that these homologous chromosomes are not identical, and they're containing different versions or alleles of the same genes. Because of this, each gamete will end up with a unique combination of genetic information. And we know that in humans with 23 pairs of chromosomes, this independent assortment creates an enormous amount of diversity. And there are going to be over 8 million possible combinations of chromosomes that can result just from this stage alone. And that's before we even consider the crossing over that took place. So by the end of metaphase 1, we're going to see that no two haploid cells will have the same genetic composition. Now let's look at anaphase 1. At this stage, we start to see that these microtubules are going to shorten, and that pulls the homologous chromosomes apart toward the opposite poles of the cell. Now notice that the sister chromatids are remaining tightly bound at the centromere. So both of these are tightly bound, both of these are tightly bound, right? And they don't separate yet. So instead, it's the homologous pairs that are pulled apart. As this happens, the chiasmata are going to break apart, and that releases the crossover connections while ensuring each chromosome is going to move to its assigned pole. If you look at this diagram, we can see that this separation is leading to different possible outcomes in our gametes. Because our homologous chromosomes carry different versions of the genes. The way they assort in anaphase 1 is helping to contribute to its genetic diversity. And this process is called independent assortment. I don't have that on the slide written, but we have it labeled for our figure. It's an important term to know. So our meiosis 1 is really generating genetic variation in two ways. We've got the crossing over that happened in prophase 1, which creates the recombinant chromatids. And then we have it through random assortment of our homologous chromosomes in anaphase 1. Not to mention, we also had that random assortment taking place in metaphase 1. That contributes to this independent assortment that takes place in anaphase 1. So finally, let's wrap up meiosis 1 with telophase 1 and cytokinesis. During telophase 1, the separated chromosomes are going to arrive at each end of the opposite poles of the cell. And at this point, the nuclear envelope starts to reform. We can kind of see that in the gray over here. And the chromosomes begin to decondense slightly, and they're becoming less tightly coiled. Next, what's going to come is our cytokinesis, which, if you remember from mitosis, is going to be that physical division of the cytoplasm. We create this cleavage furrow, and that pinches the cell into and distributes the organelles and cytoplasmic contents into each of our daughter cells. This results in two haploid daughter cells, meaning they're only going to have 23 chromosomes each in our human cells. And these chromosomes are still in their duplicated form. We can't really see it pictured here, but the sister chromatids remain attached. Now let's move into meiosis 2. So after the first division, we have our two haploid cells that can enter a short resting phase. It's called interkinesis. But unlike interphase and mitosis, there's going to be no S phase here. So in other words, no DNA replication takes place before meiosis 2 is going to begin. And the whole goal of meiosis 2 is to separate the sister chromatids. So this process will be very similar to mitosis, as I mentioned at the beginning of this lecture, and each of the two haploid cells produced in meiosis 1 goes through the familiar stages. So we see them pictured over here. We've got prophase 2, where we'll see the chromosomes condense again and our spindles will form. In metaphase 2, we're going to have those chromosomes line up on the metaphase plate. Anaphase 2 will start to pull those sister chromatids toward their opposite poles, and telophase 2, the nuclear envelope will reform, and cytokinesis will divide the cytoplasm, and we will end up with 4 haploid cells, each containing one complete set of chromosomes because of the crossing over in prophase 1 and the random assortment of chromosomes in metaphase 1 and anaphase 1, each of the gametes will have a unique genetic makeup. So I already kind of went through and gave you a summary of each of these phases. The only thing I'll mention is we did not discuss pro-metaphase 2. At this point, the nuclear envelope will be completely broken down and the spindles are fully formed, and the sister chromatids will form their own kinetochores, each where our microtubules will attach, which prepares it for metaphase, where we can line it up on that metaphase plate. And then we'll move into our other phases, which I already described, and we'll see that we have our 4 genetically distinct haploid cells. And this is just a figure that helps to summarize all of the processes in interphase, meiosis 1 and meiosis 2. I will let you look this over as a study aid. And here is a nice table that compares meiosis and mitosis, along with some text to kind of guide you through. So let's go through this really generally, and I won't describe the individual phases again, but what I want you to understand is that we are starting with a diploid adult cell. And this is going to have our germ cells, which are specialized diploid cells as well. And these germ cells will undergo the process of meiosis, and essentially we will reduce the chromosome number by half. And by the end of meiosis, we are going to produce our 4 haploid gametes. So this would look like in someone that had testes, we would have 4 sperm cells. Now, not to get into too much detail, but if this was in someone that had an ovary, only one of these would be the egg, and the others would be something called polar bodies that would disintegrate. Okay, but that egg would have the genetic information that we would need. So at this point, once we have a sperm cell meet an egg, if you remember from the beginning of this lecture, they will have their genetic information fuse. Remember, because a sperm cell has 23 chromosomes, an egg has 23 chromosomes, and when they fuse together, we will get 46 chromosomes, meaning that our zygote will have 46 chromosomes, and then this zygote will divide through mitosis. So we will be able to grow into a multicellular adult. So the whole point of meiosis is to have our organism be able to produce haploid gametes, whereas our mitosis process is used for growth and development of our organism after the zygote stage. So meiosis will reduce chromosome number, mitosis does not. It just replicates it.